Hi everybody, this is Goal 2 or Outcome 2 now, all about the cardiovascular system. There are 5 modules for the cardiovascular system, so it's pretty large. So take your time, try to do one each day until you get through them. So the first one is about the blood. So we're going to talk about the function of blood, what's in the blood, the different types of blood cells and what they do. We're going to talk also about how blood is made and degraded. And then the clotting process is part of this one, although that'll be mostly focused on the second module. All right, so blood has a lot of functions, the main one being transporting all the good stuff around the body. The glucose that the cells need from the digestive system, and then the waste taken to the kidneys to get rid of them, and of course oxygen and carbon dioxide. So lots of things are transported in the blood. It also has a buffer in it, bicarbonate, and we'll talk more about this during the respiratory system, but hopefully you remember a little bit about buffers from A&P1 that helps regulate our pH. Remember that homeostatic pH is supposed to be 7.4. The blood also contains the components to allow for clotting when a blood vessel is broken. It contains white blood cells to fight off anything that you might be exposed to when it comes to bacteria, viruses, or other pathogens. Also, because mostly it's water, and water contains a high specific heat, it makes it hard to change the temperature of that amount of fluid all at once. So it keeps us from a drastic drop or increase in body temperature because there's so much water. So think about when you have to heat up water on your stove, you have a pan of water, it takes a while to heat that up and it takes long time to cool it down. And having a big volume of fluid inside of us also helps regulate our body temperature the same way. Okay, so the amount of blood, again, in your body can be spun down in a centrifuge. And you can see here in this little centrifuge tube, when you spin it down, it separates it into the components. The main yellow part there is the plasma, which contains some proteins and any hormones or nutrients that might be around in the blood. But it is a kind of a straw colored. It's a little bit yellowish because of the bilirubin in there. And we'll talk about that a little bit later as well. There are some proteins called plasma proteins, albumin, globulins, and fibrinogen, and you should know a little bit about those, and we'll talk about them in this section. But that helps maintain osmolarity of the blood. You don't want to not have those. The bottom part of the tube is the red blood cells, and the amount of red blood cells, if you measure it with a ruler divided by the total amount of fluid all the way up to the top of the plasma there, should be about 45%. And one of the labs you're doing this week or this time during this section is hematocrit. So that's what they do. They measure, after they spin down your blood in a little tiny tube, the amount of red blood cells compared to the total, and 45% is the average. Between the plasma and the red blood cells is this tiny little white part called the buffy coat, and that's where the white blood cells will be located. Okay, a couple of numbers you should probably know. The average adult has 5 liters of blood. It's a little bit higher in men and a little bit higher in bigger people, but the average person has about 5 liters of blood. So if you think about a 2-liter bottle of pop, you have 2.5 of those. You have roughly 5 million of the red blood cells per microliter and 5 to 10,000 white blood cells per microliter. So if you have a spoonful of blood, you have quite a lot of blood cells per in a spoonful. A spoonful might be 5 milliliters, which would be 5,000 microliters. So 5,000 times 5 million is 25 trillion red blood cells just in a spoonful of blood. Again, the amount of red blood cells should be about 45%. also sometimes called your packed cell volume, and this is what they measure to determine if you're anemic. Women who have less than 37% red blood cells are anemic, and men under 42. Women are more likely to be anemic because we lose blood during menstruation. All right, the plasma proteins, albumin, globulin, and fibrinogen, again, their main job is to maintain osmolarity in the blood. They should never be outside of the blood. They're too big and are not supposed to be pushed out of the capillaries. Okay, so if you have high blood pressure and you find them in the lymph or find them in the lymph fluid, something's wrong. They also have individual jobs. Albumin help carry fatty acids and other particles in the blood. Globulins are important for immunity. They can carry vitamins and lipids, but their main job is antibodies to help with the immune system. And fibrinogen is one that's going to play a big role in the clotting process. Okay, so together, the plasma proteins help maintain osmolarity, but they also act as transporters, antibodies, and clotting factors. Also, in your plasma, you're going to find things like glucose. Okay, it depends on how much you ate that day, how much glucose you find, amino acids, and fatty acids. So, again, depending on what you ate, but you're going to find all of those nutrients. You'll also find blood gases like oxygen and carbon dioxide. Obviously, oxygen is being brought into the body to get into the tissues, and carbon dioxide produced by the tissues is on its way out. You might find some wastes. This depends again on what you're doing and what you've eaten, but always urea, uric acid, and ammonia a little bit will be found going to the kidneys to get excreted. Bilirubin and creatinine are other waste products that you might find in the blood as well. And of course, electrolytes. All the salts that you eat, remember they're ionically bound, so when you put them in fluid, like plasma, they come apart into their particular ions. You should always find some sodium, potassium, calcium, etc. These again contribute to osmolarity of the blood, and if those electrolytes include the H plus ion, the hydroxyl ion, that's going to alter your pH. So when you go and you donate plasma, which they actually pay you for, you're donating all of this stuff. They actually take your blood out, spin it down, and give you back the blood cells and just keep all the stuff, all that liquid, that stuff in there, like clotting factors and other hormones that they might want to use in cell culture or to help fix people if they need clotting factors, for example. That's how they used to get them. They often do that with recombinant DNA technology now, but you're donating all the stuff besides the blood cells. Now, the types of blood cells include red ones, white ones, and platelets. The fancy word for red blood cells are erythrocytes. The fancy word for white blood cells is leukocytes, and sometimes platelets are referred to as thrombocytes. I totally robbed this picture because it was so cool. It says all over the website, do not take. Dennis Kunkel took this picture, and I have the reference for it in the notes version of the PowerPoint if you want it. But the red blood cells here are the red ones. The little yellow things are the platelets, and that green cell there is a white blood cell. So it's a great image, electron micrograph of your blood cells. So we're going to talk about erythrocytes first. Their main job is to carry oxygen. Do not forget that's on the final exam. What is the function of a red blood cell? Carry oxygen to the tissues. Again, you have 5 million of them per microliter, and they're all carrying oxygen because they're stuffed full of hemoglobin, which binds the oxygen. They look like donuts without the hole all the way through, called biconcave discs. In fact, sickle cell anemia is when they're shaped more like sickles instead of the little donuts, and they get trapped and caught in tissues. And I'll show you a picture of that in the next slide. They tend to be redder when they're bound to oxygen and a darker color when they're not. And so when we look through our skin at our veins, the blood is a little darker, and because of the pigments in the skin, they reflect a bluish light. So all pictures, the veins are colored blue and the arteries are colored red because of this. Red blood cells only survive about 120 days. They can't live much longer than that because they don't have a way to repair themselves. They have no DNA. They don't have a nucleus. So they get kind of worn out as they get stuffed through our capillaries, and they are recycled in the liver and spleen. So they're being made continuously because they die continuously, or they get degraded, recycled continuously. So here's a picture of a capillary with some normal red blood cells, but also that sickle shape. It's because of a mutation in the hemoglobin, so it doesn't form a ring. And because of the non-ring form, then the cells form that sickle. And if the sickle cells get caught, then all those tissues beyond that point aren't going to get oxygen, and they could die, which is very painful. So the person knows that they're having a sickle attack, or a clot has kind of formed, a blockage. And one of the ways is treatment is to give that person a high amount of oxygen, give them straight oxygen, because that caused that sickle shape to pop back into the nice little donut shapes. Okay, so they look like little cinnamon candies, but obviously they're millions and millions and millions of them in the human body. So again, they have no nuclei, no mitochondria. They literally are just packed full of hemoglobin. So because they don't have a nuclei, they can't do transcription and translation. They can't make new proteins. They can't fix themselves. They also don't have any mitochondria. So cellular respiration, they only can do step 1, glycolysis. So there's not a lot of ATP being produced. So they don't really have a way to repair or fix themselves. But because they don't have nuclei and mitochondria, they have a lot more room for hemoglobin. And hemoglobin binds the oxygen. So again, the main function of a red blood cell is to carry oxygen. They don't have the ability to do some other stuff, but that's their job, is to carry oxygen. Okay, there is a great website here, or a great little YouTube about a demonstration of how blood cells are made and degraded. I highly recommend you check that out. I'm going to try to explain it here, but that website that YouTube does a great job. So the bone marrow, red bone marrow, at the end of the spongy bone, the ends of bones where spongy bone is where blood cells are made. Red blood cells are made about 2 to 3 million per second, so it's a kind of a busy place. And what's actually produced is a cell called a reticulocyte, which does have a nucleus. And as they mature, about 24 hours later, those reticulocytes become erythrocytes. What induces the red bone marrow to kick this process off is a hormone called erythropoietin. I know you're already missing the hormones from goal, or from the first outcome, from the endocrine system, but here's another hormone, just so you know you don't miss them too much. This hormone is released by the kidney when oxygen levels go down. So when the body says, oh my gosh, you don't have enough oxygen, erythropoietin is released. It goes to the bone marrow and says, kick it up, people. We need some more red blood cells. So low blood oxygen tells the kidney to release erythropoietin. It travels through the blood because, remember, the definition of a hormone is a chemical found in the blood that changes the function of a target cell. So it goes through the blood, it targets the red bone marrow and says, hey, you need to make more red blood cells. And so now you have more ability to carry red blood cells. We actually can mimic this. We can give you a shot of erythropoietin to try to induce this process if for some reason you don't make enough red blood cells. You can also induce it yourself by going to a place with low oxygen. High altitude training centers exist for this reason, because you'll make more red blood cells and they last 120 days. So athletes do this. They go up to high altitude training centers 120 days before their Olympic event or whatever they're doing. They get more red blood cells induced to be made, and they come down off the mountain, and now they have more red blood cells, so they can carry oxygen better. They have a little bit more endurance. So what do you need in order to make red blood cells besides the erythropoietin? You need folic acid, found in green leafy vegetables, and you need vitamin B12, only found in animal products. So eat those vegetables and drink some milk or eat some meat or eggs because you need both of those. Vitamin B12 is absorbed only when it's bound to intrinsic factor, which is made in the stomach, and there are people that don't make intrinsic factor or don't make enough of it, and so even though they might consume vitamin B12, it's not absorbed. So they often have to get vitamin B12 shots because you have to skip the whole digestive system. You have to get it into the blood somehow, and it's not absorbed if they don't have intrinsic factor. You also, of course, need iron. The single greatest reason people don't have enough red blood cells is they don't have enough iron. Either they don't eat enough or it's not absorbed well. Iron is actually absorbed better out of meat. The bloodier the meat, the better. Or liver, which some people can't eat very well. But you can also improve your iron absorption with vitamin C. So if you don't eat enough vitamin C, maybe your iron's not being absorbed as well. So drink some orange juice with your eggs. So when a person comes in to the doctor's office and is diagnosed with anemia, one of the things the doctor might check for is iron levels, of course, but also some of those other things on that list. The most common treatment for anemia, though, is go home and eat iron or take iron pills. People don't like to do that as much. It does make your poop turn black, and it can kind of sometimes cause constipation, but maybe the iron pills are easier than eating liver. There's a bunch of different kinds of anemia besides just the low iron one, and they're listed here. You do not have to know this for a test. Obviously, if you have anemia, the most common cause is lack of iron, but a doctor might want to check some other things if iron pills don't work. For example, do you not absorb enough vitamin B12 because you don't have intrinsic factor? Or do you have a genetic problem like sickle cell anemia or thalassemia? So all of these things might be something that your doctor might check. So 120 days, those little red blood cells are kind of getting raggedy, and as they go through the liver or spleen, they will be recycled. And the cells material is broken down, and that hemoglobin, and you have a lot of hemoglobin inside each red blood cell, is split into its two main components, the heme and the globin. The globin apart is just protein, so the amino acids can be reused. The heme has to be separated from the iron, and the iron is removed, and it can be stored in your liver as ferritin, which is why liver is high in iron, or it can be transported to the red bone marrow via transferrin. It's like a little truck that carries iron. What's left over after you take the iron off is biliverdin, which is kind of a greenish color, and as it gets oxidized and recycled down, it becomes bilirubin, which is kind of a goldish brown color, and this is part of the bile pigments, which is why bile is kind of a greenish color, and then as it oxidizes, as it goes through the digestive system, becomes brown. Literally, this is why your poop is brown, because these bile pigments end up in the fecal matter. So now you have learned why your poop is brown. Bilirubin also gets into the blood, and it's why your urine is kind of a yellow or yellowish color because of the bilirubin. So there you go. You learned something today. There is a cool web YouTube showing this process. If you want to check that out, go to the regular PowerPoints or to Canvas, and that website is there. So you should know a little bit about this process so you can answer on the questions on the test, like why is your poop brown? And what is hemoglobin broken down to? Heme and the globin. And how is iron carried to the bone marrow? via transferrin, or it can be stored in the liver. So here's a picture of a cross-section of a liver, and you can see the little bilirubin granules in the liver, which is why liver is kind of a brownish color when you look at it. Newborns have jaundice sometimes because their liver can't quite do this fast enough right when they're first born. It takes a little while for them to get all these things working, and jaundice can build up in the blood and be dangerous. And one of the ways they treat this is they put you under UV light that helps break down the bilirubin into non-harmful substances. And a nurse actually noticed this when she was carrying the babies in the nursery outside, and noticed where the sunlight hit the skin, it cleared it up. So that's why they use UV lights to help break down bilirubin now. All right, the second kind of blood cells that we're going to talk about are leukocytes, white blood cells. You don't have nearly as many of these, so when you look at a blood smear under a microscope, you see tons and tons and tons of reds, and every once in a while a white blood cell. There are 5 types, and the one you have the most of is neutrophils. I don't care if you remember these percentages, but you should remember that the most common white blood cell is the neutrophil. And that's the one they're going to look at the most as far as numbers. If you don't have very many neutrophils, you are neutropenic or very susceptible to infection, because these are the first ones that rush off everything from when you get a splinter to food poisoning or whatever. The neutrophils are usually the first one to the site of infection. So again, the word penia, leukopenia, means a low white blood cell count, which can occur because of a viral disease. Viruses, weirdly, will lower your white blood cells, even though you need them to fight off infection, which is why when you get a viral infection, you're more likely to catch a bacterial infection because your white blood cell count is down. Leukocytosis is when you have a high white blood cell count. Most often occurs when you have a bacterial infection, so appendicitis, for example. But mostly they count each one because each one has a different role, and sometimes if they know, for example, if your basophils are high, it might be an allergic reaction. White blood cells have a couple of talents that are special to them. They can do phagocytosis, which is cell eating. They wrap themselves around their target bacteria, engulf it, and destroy it. Hopefully you remember that from A&P2, a little bit talked about phagocytosis back when you talked about the cell stuff. They also have the ability to move out of the capillaries and into the tissues, and that's what this picture is showing here. There's a white blood cell in a capillary, but it can squeeze between the capillaries and go into the tissues to fight off infection. That's called diapetasis. And besides metastatic cancer cells, these are the only cells in the body that can do this. Move out of the blood cell and into the tissues. Also, if you have a splinter in your big toe, the chemicals released there because of the damage will attract white blood cells. It's like they know where to go, right? And that ability to be attracted by a chemical or repelled is called chemotaxis, and white blood cells can do that. They just know where to go because of those chemicals. So you have 5 different types of white blood cells. 3 of them have little granules in the background when you look at them under a microscope slide, and 2 of them do not. I don't care if you remember which one of these are which. However, this will help you identify them, and that's part of your lab assignments during this particular unit. Again, neutrophils are the most common. They generally have 2 or 3 or 4 lobed nucleus. You can see they're about 2 or 3 times bigger than the red blood cells in this picture here. Again, most common white blood cell is the neutrophil. They will respond to any body damage, infection, or stress and go there, and then do phagocytosis to help clean up the mess. Eosinophils have a red background. They're the only one with the red dotted background. They too have a couple of lobed nucleus there, and they tend to go up or be higher when you're fighting off worm infections. So if you have a high eosinophil count, it might imply you have a parasite. They also go up during allergic reactions. Basophils are the number one cell that is causing all the problems of hay fever, for example. Allergic reactions are caused because basophils float around and they release histamine, which causes everything from hives and itching to runny noses to vasodilation. They also secrete a little bit of heparin, which thins your blood a little. They are high during allergic reactions. They are sometimes called mast cells when they're in tissues. Basophils are the ones that are floating around in the blood, but if they've done diapetesis and moved down into tissues, they're referred to as mast cells. Monocytes are the monsters. These are the big ones. They generally don't have granules in the background. The nucleus looks kind of like a baby's butt there a little bit. They do a lot of phagocytosis, and in tissues they're called, they're called macrophages. And you might remember that connective tissue has mast cells and macrophages that move around and clean up the mess. Okay, if you remember that from Unit 4, or the tissue chapter back in A&P1, monocytes do that. And then the little ones are the lymphocytes. Again, you can see they're pretty small, just barely, maybe only 1.5 times bigger than a red blood cell, and almost all of the cell is taken up by the nucleus. You have B and T lymphocytes, which are going to play a role in immunity, which we'll talk about in outcome 3. The B cells are going to make antibodies, and T cells will become T killer cells. You can't tell the difference between a B and a T just looking at it under a microscope, but they are specific and they can last all your life. If you've had chickenpox, you have lymphocytes that will remember that and fight it off. That's why you don't usually get the same disease twice, because these cells stick around. So you have memory B and T cells as well. So think about, if a person had hay fever, you should know now which leukocyte is the most common or overactive in a person with an allergic reaction. And that would be a basophil. Okay, what chemical produced by these cells causes most of the symptoms? That would be histamine, which is why we take antihistamines when we try to treat an allergic reaction. All right, white blood cells come from the same place as red blood cells, the bone marrow. Again, those stem cells there can differentiate and make red blood cells or white blood cells or platelets. And colony stimulating factors will stimulate the release of, or the synthesis and release of white blood cells. And we can mimic this. We can give you a shot of Neulasta or Neupogen to induce white blood cell synthesis now. They do that for patients who are getting chemo who might have a low white blood cell count. Platelets, the last of the 3 types of cells, technically aren't really even cells. They're fragments of blood cells that only stick around for about 10 days, and their main job is to help the blood clot. Okay, they can also release a little bit of serotonin during when you first get cut, which is why sometimes you have a moment before it really starts to hurt. Serotonin helps give that little boost of, oh, it's fine, until you can hopefully get your hand over it or get a Band-Aid or stitches or whatever you need. Okay, in this picture, the little tiny purple dots are the platelets, so they're pretty tiny. When you look at them under an electron microscope, however, they look like chocolate chip cookies, which I just had to put this slide in here because I think it's a cool picture. All right, so now you have learned all about the different types of blood cells. One of the parts of lab this week, there are several labs for this particular unit, so get started on them, is to be able to identify the different types of blood cells. And so number 1 and number 4 in this picture are both neutrophils. Number 2 is a monocyte, really big, kind of like a baby butt nuclei. Number 3 is eosinophil, the only one with a red background. Number 5 is a lymphocyte. They're littler, round, and they generally have one big nucleus. And number 6 is a basophil, lots of bluish background with granules. And number 7 is the platelet. All the other things in here are the red blood cells. So check them out. When a doctor orders a CBC, or a complete blood cell count, they're counting all of these different types of blood cells, how many reds and how many of the 5 whites. Neutrophils, again, most common, but basophils, eosinophils, lymphocytes, and monocytes, as well as your platelet number. Okay? And it really can help with diagnosis, because if one is high or one is low, that might indicate certain or different types of problems. So there's lots of different resources for this information. You can always Google it or ask AI, can you show me pictures of, the different types of white blood cells? You also have your lab book, the OER text, the lesson, and don't be afraid to ask me. Okay, that is the end of the first module.